is so fluffy. Back to the video.
Today's Valentine's Day. I forgot. Happy Valentine's Day.
Happy Valentine's Day.
Working really well. Don't worry.
Gigi. Gigi. G-I-G-I.
So we did natural occurring variation, right? Variation must be heritable. And fitness differences, right? Yes. So what was fitness again? Remember, symbolized by W, little W. Was what? A measure of? What was fitness? It's a measure of the number of offspring that an individual produces, right? And we specifically said it's the number of offspring that themselves survive to reproduce, passing on your genes. And fitness was absolute and relative. What was relative? The more important metric. Relative fitness was what? Was what? Come on, relative fitness.
Well, it's relative towards the highest.
Right, so it's a scale value where you take the highest and divide that number into everybody else. And so relative fitness is basically a percentage of how well you do. And it's a number that ranges between 1 and what? 0 and 1. If a genotype or a character has a relative fitness of 0, it means what? Yeah, they're not gonna make it. They're not gonna. They don't necessarily die. They could, but at least they don't live long enough to reproduce, or they don't reproduce at all. And if a character or a genotype or an allele has a fitness of 1, what does that mean? They all survive. Right? The opposite of that, of relative fitness, is the selection coefficient, symbolized by little s. The selection coefficient is also a number between 0 and 1, and it's the opposite of the other one. So the relative fitness plus the selection coefficient gives you 1. So if you know 1, you can calculate the other. Right? It tells you, it doesn't mean this exactly, but it's always good to think about it. Relative fitness tells you who makes it, and the selection coefficient tells you the opposite, which is who doesn't make it, right? So if the relative fitness is 60% for a character, notice it's not 100%, what is the selection coefficient? 40%. So selection coefficient tells you selection is acting against that trait. The fitness is the positive view of that. So if we have 3 genotypes here, with these selection coefficients, okay, with these fitnesses, sorry, what is the selection coefficient of big A, big A? Against big A, big A? 0.8. What about big A, little a? What is its selection? What's the selection coefficient against big A, little a? 0.4. And against little a, 0.8. Got it? Okay, then finally, remember we had these different conditions. The last condition was not all offspring survives to reproduce. And that makes total sense, in the sense that if there's enough resources for everybody, it doesn't matter if one was faster, one was slower, there's variation, who cares? There's enough for everybody. There's no struggle for existence. So this had to be part of it. And Darwin got this from a book that he read. So the fancy way of saying all this, for all of this to work, not all individuals in a population have the same probability of survival to reproduce. And you've heard of these expressions that relate to this. The struggle for existence, survival of the fittest. Not everybody can make it. Okay? And Darwin got this from a sociology book written by this guy, Thomas Malthus. And that book was called Essay on Population. And what he was saying, among other things, is that he noted that populations, notice how the graph, this is the population size here and time, generations. As populations increase, the food supply for those populations doesn't keep up with them. What this means is that, and he was talking about human populations, right? He was saying that the human population in England was growing too fast for the resources available to support them. And what he was actually saying, therefore, the government in England should not support social welfare programs, because they won't help, because no matter what you do, there's always going to be more people who need the stuff, so let them all die. Literally, that's what he was saying, right? But Darwin looked at this and he said, this could work for natural populations, and he did some calculations. If I start off with a male and a female of this species, knowing the generation time, how many of these individuals will there be in a certain amount of time? And he calculated this for elephants, for zebras, or cows, or insects that they knew, and the numbers were humongous. But he said, but we don't have that many of this species. We don't have that many elephants. So something must be limiting them, right, in numbers. And that's when he came up with natural selection. So not all organisms, or not all individuals in a population have the same probability of surviving, but if they have a particular character which allows them to get a resource better than the neighbor, and that character is genetically encoded and can be passed on to offspring, then they will survive more, they will leave more offspring, and over time the population is going to look like those that survived as opposed to those that did not survive. Got it? Now, selection, and you're going to see this in books, and we'll talk about the salute disease. Selection can be positive or negative. So when we use the expression positive selection, that means that the individual is being favored by selection. When we use the expression negative selection, that means the individual is being hindered by selection. Got it? All right, so now, something important. I'm going to show you 3 genotypes in 3 populations with fitness values. Relative fitness. And you're going to ask you a question. You got it? And it's this. We're going to make a really important point about how selection acts. Super important. So we're going to have 3 genotypes that we're looking at. Remember, those genotypes are coding for a character. And we're going to look at the genotypes in 3 populations. We're going to call them A, B, and C. And these are the relative fitness values. Now, by looking at those values, can you tell whether or not, and you have to know genetics that we covered last semester, can you tell whether or not A1 is dominant to A2 in any of those populations? And if so, why? Or if not, why not? And then the answer to that question tells us this. That important point about how selection actually acts on populations. Give it a guess. Yeah?
Is it yes?
Yeah, but which one? If you can tell, so which
it
population
A2?
is A1 dominant to A2?
A1 is dominant to A2.
In which population? There are three populations, A, B, and C.
A.
In A, why?
Because when you have
And you have to know stuff that we talked about genetics. You can never forget that. Otherwise, you don't understand this at all. So you gotta remember what dominance meant.
because when you had at least one A1,
Uh-huh.
it was still on your what's it called? Your fitness was still on, and then as soon as you had
But what about this population then?
It's still higher than the relative physics.
So in population A, you're saying A1 is dominant to A2.
yeah.
And what about population B? No? And what about population C? Yes, also yes, but why? Because these two numbers are the same. What do you know about, if A1 is dominant to A2, what do you know about the phenotype of A1, A1, and A1, A2?
It's the same.
It's the same. And this is the point here that we're going to make because of this, and it's this. Natural selection does not act on genotypes. Natural selection does not see big A allele or little a allele. It sees you're fast, or you're tall, or you're strong, or you're weak, whatever the heck it is. So, therefore, it acts on a phenotype. Therefore, the reason these two have the same fitness is they have the same phenotype. And the only way that can happen is if this allele is dominant to this one, so this has the same phenotype as this. Alright? So natural selection acts directly on the phenotype and indirectly on the allelic frequencies. Critically important. That's what you'll be doing next week in the lab, is mimicking random mating and selection using beams. Yes.
So in B, A1 is not really because it's not the same.
Well, since these three values are different, that means these three phenotypes are different. So you don't know anything about the mode of inheritance. But we do know that this guy and this guy look the same for that character that we're measuring. That's why their fitnesses are the same. Good? Okay, now, selection acts in different ways. And we call those different ways the modes of selection. And what we're looking at is, if this mode of selection is acting on a population, how does the mean of the character change? How does the variance of the character change? And those three modes, three general modes, are directional selection, which is the most common form of selection. It's the most common way that selection acts. Then you have something called stabilizing, which has all sorts of names. Normalizing selection is the same thing. Balancing selection is the same thing. And then something called disruptive selection. Each of these, if acting on a population, will favor different individuals in the character distribution and then change the mean in subsequent generations and change the variance or not. We already did mean, variance, and mode, and all that stuff before. Good? Let's look at these. So let's look at the most common form of selection. Which is what again? Directional. So let's do this first. Let's say we start off with a population where we measure some character, whatever the heck it is, that has a mean of 5, and this distribution with that, remember, this is not equivalent to variance, but it's an indicator of variance. The wider this is, the more variable. So you go from here to here. What has happened? Well, remember, we're looking at the variance and the mean. What happened to the mean? It got bigger. What happened to the variance? It decreased. If you go from a bunch of individuals that have this distribution to a bunch of individuals that have this distribution in 10 generations, who here was reproducing? As time progressed, which individuals, remember these represent individuals, this is the frequency, individuals that have this value and that's that many, individuals that have this value, there's that many, and so on. So who is reproducing?
The mean.
The mean. No. There are individuals that have all of these values. So if you're getting bigger, who is reproducing here more than the others? At which end? In the middle? At the right, my right. The bigger ones, the bigger individuals are reproducing. This is why that's happening. But you could also get this. And what happened here? If you went this way? Huh? You got the mean change again, the variance decrease again, okay? And who was being favored? Who had the highest fitness as you went from here to here? The little ones, the smaller ones, below the mean, here above the mean. So in directional selection, the variance always decreases. And why does the variance decrease? Because if only these guys are reproducing, or not only, but more than these, you start eliminating these, so the variance, there's less of these, so less genes that code for being small. So you kind of eliminate that, the variance gets smaller. And here the same thing, because you're eliminating the ones that are bigger. So you always say that in directional selection, the mean changes because it can go in one direction or the other, and the variance always decreases. And in directional selection, individuals at one end or the other end of the distribution, let's just use the term, the smallest or the biggest, are the ones that are favored for whatever reason. They have the higher fitness. So notice what this says. So here, you have high relative fitness. Here you have low relative fitness if you're going in this direction. Let me give you a little picture. So here you have a population of plants. So same species. The leaves of these plants have little spines. That's genetically encoded. Some leaves have no spines, and some have lots of spines. A new insect predator comes in and loves to munch on these leaves. But the little spines deter it from attacking and killing the plant. So if that happens, selection is going to favor those plants that have spines on their leaves. They're going to reproduce more than the plants of the same species that don't have. They don't carry the alleles that give you spines. So over time, you're eliminating the alleles that code for no spines and increasing the number of alleles that codes for spines. So eventually, You're going towards this end of the distribution, and you're eliminating these that have few or no spines, so you reduce the variance. And you have that. Little hairs in the spines. Let's say. So let me give you the classic example of directional selection. And that is the peppered moth in England, and a phenomenon called industrial melanism. The peppered moth, Biston vitularia is the scientific name, okay, comes in 3 phenotypes or morphs. Something called typica, which is light colored, something called carbonaria, which is dark colored, and something in between called intermedia, but this is so rare that we didn't even consider it. So what do they look like? This is it, the peppered moth. This is the typica light form, and then this is the carbonaria dark form. Babies of these can be born to either. It's a genetic thing. It's a one locus, 2 alleles. Right? And the lifestyle of these guys, they're moths, so they're active at night. Okay? And during the day, they rest on trees, and they wait at night to come out and feed on nectar and stuff like that. So what happened? So in England, prior to the Industrial Revolution, and the Industrial Revolution was when they got steam engines and they got all these machines and started burning coal to make things happen, and textiles, all sorts of stuff. But prior to that, the typica form, this light colored form, was common. Within a few years of the Industrial Revolution starting and the building of all these factories and the burning of coal, this form declined in numbers and this one increased in numbers. So why did that happen? When coal was burnt in these factories to turn turbines and machines and stuff like that, you know, coal produces soot, and that stuff went up into the atmosphere. That's when we started polluting everything. And the soot will cover, in fact, a lot of diseases, respiratory diseases among the population in London and many other cities started then. But then that soot would start covering the trees. I told you that these moths, during the day, they rested on the trees. The trees had lichens, many of them. And you're going to learn about lichens in the lab. Okay? And what happened was, prior to the Industrial Revolution, the light-colored moth was camouflaged. When we talk about this, that's called crypsis. Against the background, the main predator of these moths are visually guided birds. So it's easier to see the dark one against this light background than it was to see the light. Do you see the light one in there? It's in there. Do you see it?
Yeah.
It's right here, but it's harder to distinguish. Right? So the birds would preferentially get those. So selection was against them because their coloration gave them away. They didn't eat them all, right? But they put a dent on it. The minute that the soot started covering the trees, it started darkening the lichens. So the trees, the background got dark, and now the tables were turned. These were hardly visible, but these were very visible. So the birds started eating them, okay? So therefore, their numbers declined, their fitness decreased. And we got numbers like this. So the black morph was very rare. Industrial Revolution, woo, it increased dramatically. And notice what's happening here. It's not like the light ones were turning black. It's just that they were not living long enough to reproduce and therefore not passing on their genes to subsequent to their children. So the dark one increased. But look, in the late 1900s, it's going back to normal. It's still this trend now. The light one is coming back. What do you think happened there? Right. That's when, remember in the 1960s, they started with the Green Revolution, not the Green Revolution, but the Save the Environment and all sorts of stuff, Earth Day and all that. So they forced the corporations to put what are called scrubbers. Scrubbers are these big devices. When the soot comes up, it kind of takes it so it doesn't go out into the air. And then naturally, the trees started getting back to their natural color, and it went back to the light colored trees, and so the light colored ones were protected, the other ones were more seen by the birds, and they started to decline. Natural selection. All bunch of stuff. And another example. Cipro. Did we mention Cipro last semester? Yeah. What was DNA gyrase? What was DNA gyrase? From last semester. It was what?
Cut off, explain.
No. Yeah, it's something with cutting off, but what did it do? DNA gyrase it's a bacterial what molecule? Starts with a T. I see some people on the computer not paying attention. Listen. It started with a T. Topoisomerase. DNA gyrase was a type of topoisomerase found in bacteria. And what do topoisomerases do? Right, so it moves the head of what? Of helicase. So when you were unwinding the DNA, you have to go ahead. Also in transcription, it moves the head of the DNA polymerase, RNA polymerase, right? So ciprofloxacin is a super important general action antibiotic. It worked in a whole bunch of bacteria because what it does, it binds to DNA gyrase, which is found in all of these bacteria. And therefore, they couldn't replicate, nor could they make proteins because they couldn't transcribe. And you killed them. In 1994, 86% of disease-causing bacteria were susceptible to that antibiotic. By 2000, it's even less today. I should have checked it. Only 76% of the species that were previously susceptible are not. And the reason for that is because of the gaining of antibiotic resistance. And the bacteria are now, what we did by too much use of antibiotics, we selected for individuals of those bacterial species that had a mutation that protected them from the Cipro. And that is a big issue in the world today. Physicians are mortified. And there are some antibiotics that are kept, never to be used. When antibiotics were first discovered, they were called the golden bullet. We got rid of bacterial pathogen-caused diseases. But then people started prescribing them all over the place. And all we did was inadvertently, unknowingly, select for antibiotic strains of bacteria that don't succumb, and they're spreading all over the place. So that brings us to, and that's all due to directional selection. And you have this in your notes. Okay, we are messing up stuff here with overuse of antibiotics because of the reasons that I just told you. So how can we reduce that antibiotic resistance? Well, first, antibiotics work, and people used to do this all the time. You came in there with a cold, you came in there with the flu. Here, take these pills and call me tomorrow, whatever the heck it is, right? So don't use antibiotics to treat viral infections. Antibiotics are molecules produced by bacteria and other organisms like fungi and plants to kill bacteria. They don't work on viruses. Avoid mild doses of antibiotics over long periods. Why? Because if you use only a little bit, you kill some of the bacteria, but some of the other ones may have time. Remember, these things reproduce every 20 minutes. You can start off with a few bacteria in your body, have billions within a day, right? So if you just only kill a little bit of them, then some of them may have the potential to gain resistance by a random mutation. And we do this all the time. When treating a bacterial infection with antibiotics, take all your pills. The minute you feel good, you stop taking them. Because, oh, I want to go back to drinking, or whatever the heck it is. You should follow the same course, because what you have done is you may have killed the majority of the bacteria that is causing the illness, but you still have some left. And the minute you stop taking it, they may come back. Use a combination of drugs, and that's what they do. A lot of doctors now are prescribing more, depending on the severity of the infection, more than one type of antibiotic, because if you have individuals of a particular species in you that may be immune to one of the antibiotics, then rarely will they be immune to both. Okay? And reduce or eliminate the prevented use of antibiotics on livestock. Believe it or not, the majority of antibiotics produced in the United States don't go to people, they go to livestock, cattle, sheep, and poultry. And the reason is not necessarily to prevent disease in them, although that helps. It's that for reasons that are still not 100% known, giving antibiotics to livestock makes them increase in weight, in volume, so you have more meat to sell. Okay? But as a incidental byproduct of that, a lot of bacteria which may be present there may gain resistance because you're overusing this thing. So very important. You have this in your team. Another example of directional selection. This is a experiment that's still going on at the University of Indiana or Illinois, one of the two. And what they did was, in the 30s, they took corn and they noticed that there was variation in the amount of oil in the little corn kernels. Okay? So they said, you know what, let's breed the corn plants that have high oil with each other and the ones that have low oil with each other and see how far we get. And over a bunch of generations, you got to the point that you had no oil, but you couldn't go beyond that. Once you got to no oil, to this day, they're still producing plants that have huge amounts of oil. There's so much oil you can literally take a kernel and light it up, right? And that hasn't stopped yet. So over here, the variation ran out, but over here, the variation is still there. Right? So they're still working. That's directional selection. And this is another one. This is manipulation of mice for size. This is the ancestral population. And they took the biggest mice and bred them with each other, and only in a few generations, they got that. This mouse was a direct descendant of these mice. Okay? And that's directional selection. Another one. But this is not natural selection. This is artificial selection. Believe it or not, cabbage, Brussels sprouts, kale, broccoli, cauliflower are all the same plant. Same species of plant, Brassica oleracea. And what you're doing is selecting for different parts of those plants. So the cabbage are the terminal buds. So you'll be learning about things like that in the lab. The Brussels sprouts are the lateral buds. These guys have nice lateral buds. Let's breed these plants that have those lateral buds that are different from the other lateral buds, and you keep getting this. Kohlrabi for the stem, kale, modify leaves, broccoli, stems and flowers, and then cauliflower, just clusters of flowers. And because you have variation in different individuals of this, and they noted that, well, let's breed these guys with these guys and see what we get. And that's how you got all these. And that's, all this artificial selection is directional selection. We'll give you this, we'll give you that, we'll give you this. The other form, another form of selection, is stabilizing selection. Remember, this is also called normalizing or balancing. So what's happening here? What's happening there? The mean is staying the same. What happens to the variance? It's decreasing. Goes from here to here to here as long as the scale is the same. So what's happening? Who is being selected for? Who has the highest fitness? So individuals with the middle value do better than individuals with either extreme. Remember in directional, either these guys were favored or these guys were favored. Now you're favoring the individuals in the middle, and you're selecting against these guys and these guys. So the mean stays the same, the variance decreases. And now this has actual medical relevance. I'm going to see right there. You got this? Yes? So stabilizing selection, believe it or not, is one reason that some recessive genetic conditions, all genetic conditions are rare, right? But some of them should be rarer, and they're still around. And one reason they're still around is stabilizing or balancing selection. Okay? And they're found at high frequency. And the classic example is, did I put it here, is sickle cell anemia. We talked about sickle cell anemia last semester. Remember, sickle cell anemia is a genetic condition caused by a recessive allele. At a locus that has two alleles, let's just call one big S and one little s. The big S is dominant over the little s. So you have three genotypes. Big S, big S, big S, little s, little s, little s. Sickle, if you're little s, you have sickle cell anemia. If you're big S, you are unaffected. I hate that term, normal. You're not abnormal if you have this. You have sickle cell. If you're heterozygous, you don't do as well as these individuals under certain conditions, and you have sickle cell trait. So what happens here? This affects hemoglobin. Tell me about hemoglobin. Hemoglobin is what kind of macromolecule? It's a protein. It's what type of merrick protein? It has quaternary structure because it consists of more than one polypeptide. How many polypeptides? Come on, you got all this stuff you can't forget. You know as doctors. What merrick is it? How many polypeptides does it have? 4, so you are? Tetrameric. No, quaternary structure means that the protein consists of more than one polypeptide. If it consists of 2 polypeptide chains, it's dimeric. If it's 3 polypeptide chains, it's trimeric. If it's 4, it's tetrameric. And that protein is coded for by 2 genes. Two of those protein polypeptides are called what? See? You can't. You think you're in high school. You cannot forget. You want to be doctor for heaven's sake. In a year, you're going to be doing this crap over there. You can't forget it. And you're not going to remember it there either. You got to remember forever. You chose this path. Your buddies at the American school all partied. You chose to be in medicine in 2 years. So you gotta work hard. So how many? What are they called?
Second, repeat question.
What are there are two types of polypeptides that make up the tetrameric
It's the alpha.
hemoglobin.
Alpha and beta.
Alpha and beta globins, right? So don't be afraid. One polypeptide is called alpha globin, and the other one is beta globin. 2 alphas, 2 betas. They're coded for by 2 genes: the alpha globin gene, which is on one chromosome, and the beta globin gene on another chromosome. Then they're transcribed, then they're translated, and then they're put together. Okay. Remember, it's a tetrameric protein, 2 beta chains, 2 alpha chains. And this affects sickle cell, the beta globin. And we call a hemoglobin that has the abnormal beta globin polypeptide hemoglobin S, and the normal one is hemoglobin A. Okay? And what happens here? Nothing under normal circumstances. So each red blood cell, that we'll learn this semester, has about 250 million hemoglobin molecules. And that's how much oxygen you need, right? How many oxygens bind each hemoglobin? That you should know. When a hemoglobin molecule is fully saturated, how many oxygen molecules are attached to it? Once again, this is stuff, physiology, to help you save people's lives. You have to know that. How many?
Is it
4,
4?
right? 4, yeah, don't forget, that's fully saturated. So anyway, when these hemoglobin molecules are filling up the red blood cell, and the red blood cell will look something like that. It's concave shape. Under low oxygen, high acidic conditions, and we're going to talk about this when we do physiology this semester, that happens in your tissues that you're respiring. The inside of the cell becomes acidic. And there is when you run into a problem, because that mutant hemoglobin, which literally just has a different amino acid at one position, the 6th position, you'll learn that too. When it becomes acidic inside that cell, that hemoglobin changes shape and it sticks to every other hemoglobin. And the whole cell collapses and gets this weird shape, right? And then this can carry oxygen. Remember we mentioned that. So that's anemia. That's the anemia part of sickle cell. You're anemic. You're not getting enough oxygen. You're lethargic. Okay? You're not, your cells are not, your organs are not functioning properly. You're not getting enough oxygen. But also, remember this was pleiotropic. The membrane becomes kind of sticky. And as these cells go through capillaries, which is always one at a time, we're going to see this semester, they can stick to each other and block blood flow to organs, resulting in even more damage. So sickle cell anemia, prior to medical intervention, had a fitness of 0. What does that mean? What does that mean? If you're homozygous for sickle cell, you had a fitness of 0. You died before you reproduced. Now with medical intervention, and even though we have not conquered this thing, people can live long lives, even though some worse than others. Okay, and that's a big problem in the Caribbean. So, what does this have to do with balancing selection? Well, if an allele has a fitness of 0, you expect its frequency to be what? 0 or close to 0. And that's exactly what happens in most parts of the world. Except you go into parts of India and Africa, especially Western Africa. You also get it in some parts of the Mediterranean. And in Western Africa, holy moly, 14%, 0.14 for an allele that has a fitness of 0. selection of 1, selection coefficient of 1. How the heck does that happen? Well, people started looking at that, and they noted one thing, that the condition correlated with the presence of a disease called malaria. And they noted this. In areas without malaria, if you were a homozygous, unaffected, big as, your fitness was 1. The sickle cell trait, this is prior to medical intervention, was near 0, and SS was 0. But holy moly, in areas with malaria, the tables were turned. Once again, if you had sickle cell, you died of sickle cell. 0 fitness. If you were big as, your fitness was no longer 1. It was really low. Not 0, but low. And if you were heterozygous, your fitness was 1 now. What the heck? And the reason was this. In areas with malaria, individuals who had sickle cell still had sickle cell, not good. If you had this, you died of malaria. But if you were heterozygous, you lived longer than these guys and lived long enough to reproduce to pass your allele down to your kids. Okay? And why were these guys better off? Not great. Why were they better off in areas with malaria? Because the malaria, when we're talking about malaria later, the parasite that causes it, it attacks red blood cells. It eats hemoglobin. Okay? And if you were heterozygous, you got sickle cell trait. That means some of your cells sickled under certain conditions. And if the parasites were inside, what do you think happened to them? They die. And what happens to your parasite load? It gets reduced. And what happens to your likelihood of surviving? It increases. You're not great, but you're better off than these guys who are dying of sickle cell, and you're better off than these guys who are dying of malaria, and you live just long enough to pass on your allele. Okay? And this is a form of balancing selection where the middle value does better than either extreme, and this form of balancing selection is called the heterozygote advantage. The heterozygote does better. Or another synonymous term is overdominance. Those two things are the same. Yes?
Are you explaining it why the carrier is more likely?
Yes, because these guys, they have sickle cell trait, right? It's not good to do that. Look over here, right? Prior to medical intervention. But in areas, the parasite attacks red blood cells. These guys, all their cells, red blood cells sickle. Here, some of them sickle. It's random. So therefore, if the parasite is inside and it sickles, it kills them. So you reduce the number of parasites in your body and live longer. Long enough to find your girlfriend or boyfriend and get married and have kids. And pass on your allele to other generations. In fact, it is believed that, we'll be talking more in semester 4, but there are some blood disorders called beta thalassemia, alpha thalassemia, the Duffy gene, the glucose 6-phosphate dehydrogenase deficiency. All of those things we think exist because they protect against different forms of malaria in the heterocyte. And this brings up a point here. Okay, and it's this. Selection is a contingent process. Okay? What that means is that what's good in one environment may not be good in another environment. Contingent means dependent on. All right? So notice that there's no absolute value for something. The little s allele In areas without malaria, it's not good. But the little S allele, if it's combined with a big S allele in areas with malaria, is good. It's dependent on the environment. Okay, let's put this. Got it? We're gonna see, when we finish disruptive, we're gonna see that selection for and against certain alleles is the reason we still have some genetic conditions in the plant or affecting us. Got it? Okay, we'll leave it at that.